back up to our workflow up at the top. And we're down here in the workflow. So we just got done quantitating our aligned reads with RSEM. We saw that we got two quantitation output files with RSEM, one ending in genes.results, and that's showing us quantitation for gene IDs from our reference index that we provided, and also isoforms.results, which gives us isoform level quantitation according to the isoforms that are annotated in that annotation file that we use to generate the RSEM index. And then just before we left, we went ahead and compiled the summary log files that RSEM produces in a file that ends in CNT. And looking at the multi-QC data, if we want to jump back to that, it would be step 8B of our workflow. We went ahead and took a look at the output for the HTML file for RSEM, and I attempted to have you guys answer some questions, but we had some difficulties with the slides being brought up. And so I just went ahead and went over the answers to both of these questions. So this is just asking which sample had the highest percent of alignable reads that were quantitated and which sample had the lowest. And then the second question was reverse to that, the sample that had the highest percent of unalignable reads, and then the sample that had the lowest. And I just let you guys know that those answers can be found by going to the top of the Count multi-QC report and looking at the alignable column. And again, you could toggle and sort the data from highest to lowest or lowest to highest. And we saw that ground control rep 2 had the highest percent of alignable reads that were quantitated at 90.5%. And that ground control rep 6 had the lowest, although not too far away, at 87%. And then to get the samples with the highest unalignable reads, you'd basically just do 100% minus this value to give us 13% unalignable reads for ground control rep 6. And then 100 minus 90.5 to give us 9.5% for unalignable reads for ground control rep 2. Lisa, that would be great when we start. So Lisa just let me know that the Menti link should be working again, so we'll use that again when we answer questions after we generate our raw counts table. And so I am still waiting for 2 people to get back. If you got back from lunch and you haven't put back in the chat yet, please do so that I know that you're back. There's one, I think we're waiting on one more. So Lauren, just a heads up, if I think we're missing Ty. So if Ty gets back as we're in the middle of something, I may have you hop into a breakout room to catch her up. So moving on now, are there any questions about RSEM, how RSEM does its quantitation, interpreting the multi-QC report for learning how many were alignable? Okay, great. If you guys do think of a question, just feel free to interrupt me or shoot your question in the chat, and I'll stop and answer it. So now that we have the genes.results files from RSEM for all of our samples, so we have a quantitation of all of the genes that are annotated in our reference for each sample. The next thing we need to do is compile this information into one table containing the expression, so the raw counts value, which is going to be taken from the expected count column of the genes.results file. So that 5th column, the expected count for genes.results file is going to be the raw counts, the number of templates that align to each gene. And so we're going to compile that information into a table. So I do want to point out that usually in our standard workflow, we actually pull in the information in the genes.results files from all samples using a program in R through Bioconductor called tax import. And the reason that we do use the tax import is because it's a function of it works with Day6 2. It makes data in the format to where we can easily create Day6 2 dataset objects, which Lauren is going to go over with you. but also it'll look at the expected length of each of the genes, and it'll normalize the data for gene length when you pull it in with tax import. However, because that is in our program, in the kernel that we are running right now is currently Python, we're going to instead use a Python script that Lauren wrote to compile all of the expected count values for all of our samples for each gene in the genes.results files into one table. And so this script here, again, written by Lauren, I'm not going to go through each one of these in details. This is written in Python. So those of you, I know that many of you are not familiar with Python. It's not our intention to teach Python right now. And as I mentioned, we usually just pull these in through tax import, but because we wanted to end this notebook by putting everything together into a table, Lauren went ahead and created this Python script, which is going to pull again. Ty, I know you just joined, so you didn't miss much. I just went over what we had ended with, interpreting the RSEM multi-QC report. And now we're going to compile all that information into a raw counts table, which is ultimately going to look like what I showed you all in the lecture. So we're going to pull in that expected count value for each gene ID for each one of our samples into a matrix like this. And again, Lauren wrote a script to do this here. Normally we do this in R using a program called tax import. And so again, not going to go over the code here in any kind of detail. If you guys do have questions, you can feel free to ask and I'll pull Lauren to answer your questions. But so we can go ahead and run this code block. And it shouldn't take too long to run. And all that that's doing again is it's just taking a look at the genes.results file for each sample. And for every single gene, it's pulling the expected count value in that fifth column and just putting it into a table. And so is there anyone who was not able to run the Python code block? Module not found error. Hey Joe, what will are you seeing Python conda environment GL4U idle in your bottom left?
Yes, I am. Yes, I am.
Go ahead and go up to the top. You can use your table of contents here if you want, and just rerun the variable definition block again.
Yep, doing it now.
And then you can jump back down to step 9 here and try running it again.
Yeah, still hitting the error. No module called pandas.
Can
It's okay.
you go ahead and share your screen?
Yes. Where is that option? Share.
Oh, so you're in the
I
wrong
think
conda environment. So
Oh, I got the wrong
see how
one.
your
Oh,
GL4U-R? Yeah, so just go ahead and click that in the drop-down menu. You want the second one down? So that one's
oh, yep.
yeah, you want that one. Go ahead
Yep, my
and
bad.
select.
Yep.
That
All good.
connects. Once that connects, run that variable code block. So go to the define paths section. You can find it in your, yeah, so the define paths just below that. Yeah, run that and then you should be able to run.
Oh my God, sorry.
No,
I
that's fine.
closed it during the break. There we go. Thank you. My bad.
Yeah,
How
no
do I
problem.
share?
And then just make sure it should have ran now. Great.
Yeah, it's ran. It's ran. Thanks.
It's okay. I love it when it's a problem that's easily resolved. Is there anyone else who's having trouble running this code block here? Okay, great. So basically, again, what this code block does, just compile all of that expected count information for each gene for every sample into a table. And then the last command that you see here in the script, it's going to write out those raw counts or unnormalized counts into a CSV file, which we're then going to be able to pull in at the start of the R notebook. So let's take a look at the first few lines of that table that we just put together, and then we're going to answer some questions. So go ahead and run this code block here, and this is a Python command as well that's just calling that counts table that was saved in the script and looking at the first few lines of it with the Python version of the head command. And now that we have this pulled up, you can see that this looks like the table that I went over in the lecture. So we have our gene IDs, again, gene IDs for Ensemble because we pulled it from the genes.results file. And then we have each sample as a column here. So 6 flight samples and 6 ground control samples. You should, if you click into where the table is, be able to scroll if you can't see everything. And so we see a certain number of genes listed. I'm not going to count them for you since that's one of the questions. And then this is going for each one of those genes, we see the expected count value from RSEM genes.results file for each one of our samples for each respective gene. So I'm going to have you guys take a look at this table and start answering some questions. So Lisa, if you could pull up the how many genes are shown question. And maybe let's put the link in the chat again. I think I still have that link up here. Oh, thanks, Lisa. So yeah, go to the Mentimeter link and you should see the question, how many genes are shown. So we'll give you a few seconds to count the number of genes that you are seeing in the head of our counts table here. I'm going to go ahead and view everyone's responses. How many genes are shown? So a lot of right answers, one Roman numeral. Yeah, you guys are right. We got 5 genes shown, and you could tell by the 5 unique gene IDs listed in this first column. Seems like all of you got that one, which is great. And so we have this next question here. Lisa, if you want to bring it up, which of the genes shown are not expressed in any sample? So which gene is not expressed in any sample? Any of the 12 samples? Awesome. Okay. Let's take a look. Everyone says the gene ending in 3. Ones that understand why it's being shown. Okay, great question. So yeah, you guys who said the gene ending in 03 is correct. So this gene is not expressed in any sample. And the reason that even though it's not expressed in any sample, it's being shown, we haven't done any filtering yet. And so what we did in the RSEM is RSEM just looked at every single annotated gene. So basically every gene that has an ensemble ID and coordinates on the respective FASTA file. And it just looked to see if any templates were aligned to that gene. And so that's what it's reporting. For every single gene in the annotated file, for each sample, was there any templates that align to that gene? So if there is, it'll tell us how many were aligned there. If it's not, it'll say that none were aligned there. So we're just looking to see, right now, this is our raw counts data. So we're just looking at every single annotated gene we have, how many templates were found from each sample that mapped to that gene. And so we haven't filtered this at all. We're just telling them to report it. Does that make sense? Or is that so confusing? Okay. That is a good point though, because it is kind of unnecessary to have genes that aren't expressed in any sample. So we are going to get into that when Lauren walks you guys through the differential gene expression. So I'm glad you pointed that out. So our very last question of this Jupyter notebook, Lisa, if you want to pull it up, which sample has the most reads that align to the gene ending in 28, the one we've been looking at, and which sample has the least? So take a look at the table and tell me which sample has the most reads aligning to the gene ending in 28, and which sample has the least reads, or I should say templates, aligning to gene 28. How are we doing on responses, Lisa?
5, 6.
Just looking at this gene 28.
Okay, we have 8.
Good. Okay. So I'm seeing a lot of most flight rep 5, a lot of least flight rep 3. Yes. Oh, this someone was looking at. I don't know what you're looking at here. So, whoever wants to answer, if you want to explain what you're thinking, though, if you're comfortable. If not, that's okay. So you have a few different genes listed here. So the question here is which samples has the most reads, or I should say templates, so I'll correct this in the notebook, aligning to just one gene. So we're just looking at the gene that ends in 28, and we want to know which sample at this point has the most of the templates aligned to that gene. And so the only gene we should be looking at for this question is gene 28 here. And then we're just going across, oh, go ahead.
That was me. I misread the question. Sorry.
Yeah. Okay. As long as it's clear now. So we're looking at this gene 28 and seeing which sample has the highest expected count, essentially, for this gene. And what a lot of you guys noticed is that flight rep 5 at 193.0 has the most, it has the highest value here for this gene. And then you guys also noticed for the least that flight rep 3 at 87 has the least amount of templates or expected counts for this gene. That brings us to the end of the notebook. We now have a raw counts table or an unnormalized counts table showing the raw expression of all of our annotated genes for all of our samples that we can then start to use to do differential gene expression analysis. So just to quickly summarize everything that we went through in this Jupyter notebook, I know it's been long and a lot of new concepts for just about all of you. So we started with our raw FASTQ files, our raw reads. We took a look at the quality of that raw sequence data. We noticed some low quality reads and some adapters, and so we trimmed those adapters off and filtered for quality, and then we did another QC assessment and saw that we were able to remove all of the adapters and all of our quality was really high once we did the quality trimming. We then went ahead and aligned those trimmed reads to the reference genome using STAR, which first required a STAR index. And then we took a look at those alignment files and we took a look at the log files showing how well, or not, but well for us, the alignment went, compiling all of our information in a multi-QC report. We then indexed those aligned reads and also converted our GTF annotation file into bed format, both of which are used to evaluate the strandedness with the RCQC and FIR experiment. We were able to identify that our reads, which were prepared in a stranded library prep kit, were stranded, and they were stranded in the antisense orientation relative to the reference. And we were able to view all of those together using multi-QC of that infer experiment output. Then knowing the strandedness, we were able to go in to quantitate our aligned reads using the align to transcriptome BAM file. So we were looking at our templates that were aligned to transcripts in RSEM. And again, RSEM required us to create that index of the reference. And then from that, we were able to view the raw count data for each of our sample for each gene in our reference genome. And then we took a look at the quality of that quantitation and noticed we had really high percent alignable and quantitative reads for each of our samples. And then we compiled all of that raw count expression values into a table. And we have ended this notebook with a raw counts table containing expression values for every single gene that's annotated in our reference for all 12 of our samples, 6 flight and 6 ground control. Questions about anything we went over in this Jupyter notebook or the respective lecture material? Anything anyone wants me to go over again?
So Amanda, this is Elba. Okay, so this is great. Now we were always at the end. I'm trying to be consistent here, just because it's going to help me organize things after this is over.
Mhm.
So in parentheses at the bottom, these were other programs you were reaching out to and implementing. But we were always in Python, in the Python environment, I thought.
So
Were we not?
we
In
were
the
in the
notebook?
Python environment. We were actually running Unix commands within the Python environment.
Correct.
So if you'll notice, all of the commands had an exclamation
Yeah.
point. So we were technically
Right.
running
Right.
everything within the Unix system. And so all of these tools are packaged so that they can be called within a Unix system. And so these tools are actually written in a variety of different languages. Some of them have some parts also written in one language or another, but they're all wrapped and packaged to be able to be called through a Unix-based system.
Okay, so then why is the last one labeled Python specifically?
So
The
this
raw counts.
So this, we didn't use any prepackaged program to compile the
Okay.
information.
I
So
see. Okay.
what we normally do is we actually use a program called tax import to compile all of our raw sequence data, or all of our raw count data, excuse me. But because tax import is an R program, and we really wanted to end this notebook by putting everything together, what Lauren did is she created a custom script here. So Lauren wrote this custom script in Python, was the language that she used to write this script, to be able to essentially pull that expected count column for the genes.results file for every sample, for every gene, and put it all together in the final table. So this isn't any program that is already made. This is a unique script that Lauren put together.
So the last thing was a script by Lauren, but all the others were Unix based that are then operating within this Conda environment.
Yeah, they were called within a Unix system. They could have been written in any number of languages, and they are written in any number of languages. Some of them are written in Java, some are written in Python, some are written in C++. I mean, they're a variety of different languages that are used in these tools, but they're all wrapped and packaged to be able to run on a Unix system.
Great. I mean, if it isn't too much trouble for me, I would love to see those, just for the sake of completion, star use, blah, 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 and blah, RSeq. I mean, it just helps me to have the level of detail that I need to be able to explain things if I ever teach this. And I need to be a lot more confident in my understanding of what's happening beneath this code to feel comfortable being in front of a class myself.
For sure. All of these codes, I think almost all of them are open source. So
Okay.
you could actually view the source code for, I don't know if anyone,
Okay.
are there any of these tools that we use that don't have their code as open source on GitHub? And in the documentation, so throughout the Jupyter Notebook, the first time I use a tool, I always have a link out to the documentation for that tool.
Great. Okay.
So in the documentation, it'll tell you what language.
Okay, so I can do that myself then. I could just go through it systematically and annotate that slide.
Sure. Yeah, absolutely. Yeah. So what's in parentheses here are the tools that were used to do each step. Yeah, but yeah, the tool documentation will tell you which languages those tools are written in. And like I said, some of the tools were written in a few different languages for different parts of the tools or different features of the tools. And again, the developing world is just so collaborative that sometimes some tools, someone will, write an add-on to a tool in a certain language, and it might not be the language that the tool is written in, so they'll wrap their tool to be compatible with the rest of the module. So yeah, that's more like heavy development computer science side, far from my area of expertise. So if anyone else that are better developers than I am knows the languages for any of these, feel free to chime in.
I did want to comment on, I don't want to mischaracterize how you're thinking about things, although maybe I misheard or misunderstood. I don't think there should be that much worry about understanding the languages and reading the code. I think understanding the documentation and how the tool is used is where we should focus, unless we're trying to learn programming and practicing programming. Aside from that, yes, there's a level we have to trust that it's doing what it says it's doing, but it's also where, like, running test data sets and inspecting, we should always inspect our outputs and never assume anything works perfectly. But just to clarify sort of for everyone, I don't think it's expected that any of us should know the code inside out for any of these tools, but more the documentation is where we should focus. Just my 2 cents on that.
Yeah, I agree. I mean,
So you bring up the issue of trust, Mike, and I have issues with trust.
Oh
And
yeah.
so and so I just need confirmation of some way. So I imagine that when you work with this, you have a file that you know what the outcome should be, and you check these programs with it because I mean these are huge. How do I know it's right? How do I know it didn't skip a column? How do I know it didn't, move something around from a file and assigned it to something else? How do
Yeah,
you
this
know
is
this?
all super uncomfortable. And honestly, there's no world where you get comfortable enough with every language to be able to assess things at that level, for any normal human being at least. Um, like you said, being able to have a test data set and being able to know what you expect and to spot check things afterwards and make sure it makes sense to you biologically if it can, all those things need to happen all the time. Every code base can have bugs in it, and they do, and we catch them and we fix them, and that's how we progress. But, right, I agree, we don't trust it, but we have to trust things to an extent, is kind of what I mean, I guess.
Now, I imagine experimentally, you can also do things to samples. Like when you do microarray, you spike the cocktail, you add something to it. I'm learning lipidomics, and so there's things you can do that you then know to some extent what you think you put in chemically, and you can confirm that there was an output. So presumably, that also is a component of experimental design here.
Mhm.
yeah, and that's a good point. And like I said, for instance, I don't know off the top of my head all the languages that are used in all of these tools. I don't know the details. I'm far from a developer. But reading documentation is really the most important in knowing and understanding. And we do and often do use things such as, so there's a variety of different controls that we use internally. We also have one of my colleagues, Jonathan, has been doing a wonderful job at putting together a validation and verification automated program to go through and do automated checks with the outputs on each file to double check things like when you compile everything into a table to make sure that it's associating the right values with the right samples from the original genes.results files, for instance. So we are soon going to be releasing that V&V program that anyone could use to check their data. We're also, Jonathan's been putting this all together in a nice workflow, so we could literally just type the GLDS number and a couple other bits of information, and then have it go and run from start all the way to finish, even what Lauren's going to go over for the DGE, and do the validation and verification steps after each output and actually halt the program if it runs into an issue, if it doesn't pass a V&V check. The other things that we have for controls is every time, at least in-house at GeneLab, when we sequence RNA-seq samples, we always include a universal RNA reference sample. And so there are universal RNA reference samples that are available. There's at least I know one for human and one for mouse. And so we usually, for our mouse studies, will include a universal mouse RNA reference and take a look at the expression of the genes in there, because we have an idea of what the expression of those genes should be if they're well documented. We also include ERCC spike in. So ERCCs are synthetic RNAs that you can spike in right at the beginning of your library prep when you do your RNA extraction. When you have your extracted RNA from your samples, we actually spike all of our samples with some consistent amount of ERCC genes, again, synthetic genes that have a known concentration. And then for those ERCC spiked genes, they go through this whole process, and then we look at the expression of those ERCC genes compared to the known concentration. So we have essentially, and I'll pull an example up in the break, but we have essentially the known concentration of the ERCC gene and how many counts was provided after we do the quantitation to make sure that it's scaling appropriately. So that way the ERCC genes with higher concentration have more counts than those that don't. So there are a whole bunch of controls that you can do and can use. All of them have pros and cons, of course, as well. So there are a variety of things that you can do to help control some of these steps, and we do those at GeneLab. Of course, this is already a lot of information, so I didn't want to continue to overwhelm all of you by adding in all these additional QCs that we do internally at GeneLab to make sure that we're confident in the data that we're providing to our users, especially those from the samples that we sequence in-house. But I also even did mention some of the RNAseq QC analyses that we do that I didn't go over with in detail here, again, just to make sure that we're seeing consistencies and things that we expect in our data. So yeah, that's a very good point. I'm also untrusting and still new to the programming world. So I have spent way too much time checking and rechecking things like manually, and then finding out piecemeal codes or asking my colleagues to write some codes to do checks. And they're like, why? It's always going to do the same thing. I'm like, no, but we got to check. And so I know that it makes them frustrated, but I'm just like you. I don't trust anything. So
I want to put one more underscore into this whole thing that's sort of, it's so important that I turned my camera on for it. This is also why documenting our code exactly is so essential. Whatever we get at the end, it's never the quote unquote truth. These are many foggy windows stacked on top of each other. What we get is based on what we did. And so showing exactly what we did allows anyone in the world and us to revisit and make sure things are the same in the future and find problems that could have been there before. It doesn't negate the responsibility of the researcher to make sure things are right, but it does help with that load a lot. This is another thing in my like 5 things I wish I knew when I started bioinformatics. We have to make decisions all the time about what we're doing, and there's not always a right and wrong at all. And so recording what we did, documenting it, making it open, that's how we sort of protect ourselves. We're being completely transparent. We did the best we could at the time. It might be wrong, but these are all the reasons we did what we did. So that kind of leads to, we're not trusting it to be right. We're trusting that it's doing what we think it's doing, and we know it's wrong, but we're trying to pull out what we can from this data.
and that's
Yeah,
also why we consult with so many other scientists. That's one of the reasons that we established our AWGs, because there are a million different ways to do each step of this pipeline, a bunch of different tools that can be used. And we don't trust that we're not going to be comprehensive experts on every possible tool out there. So the more people we get involved, the more input we get, the more confident we can be that when building a consensus pipeline, it's the best option for our data at the time in which it's implemented, according to as many scientists as we can have their input in.
Yeah, I'll also add on to what Mike was saying and mention the importance of versioning in terms of not only documenting your code, but documenting the database version that you used and the version of the program that you used.
Yeah, very good point. And I'll go over our repository in a second, but I do just want to address some of the questions in the chat. So Ty asked, how do you get the needed tools and kernels into your console terminal to be able to use them? So Jupyter Hub works nicely where you can create environments through Conda. So Anaconda, or the shorter version being Conda, is what a lot of bioinformaticists use to install tools in kind of their own isolated locations. Again, because these tools, because they use a variety of different languages and different versions of those languages, depending on what the tool is, we can have incompatibilities if we try to install all of the tools that we're using in one place, because some of them might use the same tools as others, but different, or the same languages as others, but different versions of the languages. And so we create little environments through Conda to be able to do that and install our tools. And so what we did here is we had our POCs for the SMCE system that we're working on. We put together a list of the tools that we wanted in our environment for this Python version for the fast queued accounts. We said, hey, we need all of these tools installed, this version of the tools. Here's a file to be able to use Anaconda to install it through the Jupyter Hub to be able to run. And so that's why when you're going through and selecting these environments, they have certain tools in those environments. So as we just noticed recently with Joel was using this environment, which wasn't the environment that we had the tools needed for this to run this Jupyter notebook. So he wasn't able to run things because it was looking for tools that didn't exist in this environment. So that's why we want to make sure that you're selecting the correct environments for each one of your notebooks. And that is described in the GitHub. So that way you guys know when you're putting this on. But yeah, we have our POCs at SMCE install that stuff because we're using their system, right? So they're not gonna let us install things globally into their system. So we tell them what we want, and then they work it out for us, and then we open things up, test it, make sure all the tools we expect to be there are there, and then kind of give them a thumbs up or let them know if we need to add any tools that aren't there. So if there are any additional tools that we don't have in here that you tried to call that you want to see, let us know, and we can ask our POCs to create an instance to help you when you teach at your home institution. I think the rest of the comments are just about the environments. Any other questions about this, or else I'm going to stop sharing my screen and we can go into our debrief section, which I know we kind of already started. Okay, I'm going to stop sharing my screen for now. And you guys are welcome to turn on your cameras if you want to, unmute your mics. Now is the time where I want all of us to kind of talk about and debrief on this RNA-seq overview lecture, at least until where we are now, which is through RSEM to combining these into an expression matrix. We are at 1:30. Thank you guys so much for the feedback on this first section. So I'm going to let you guys go and take your 20-minute break. And when we get back, we're going to finish up the we're going to finish up the RNA-seq overview lecture. And then you guys are hopefully, with time allowing, going to get started with Lauren on the next notebook. So please be back here by 1:50 p.m. Big time.